Now I'm going to solve some questions. Hopefully they can be useful for you to study for the test. So let's start with the first one. So here, let's read it together. The spindle pole. So here they are talking about, all these structures where there's microtubules that help to pull the chromosomes. And the spindle pole is organized around a small region. Yeah, so I want that you start making a picture. So here you have the spindle pole. Here you have, remember, here is the spindle pole. Yeah, all these structures. Um, and here there will be the chromosomes. Yeah, and they are asking you for this small region that is located here. Let me put it in blue. Yeah, so this small region that is close to the nucleus, and they're asking you what is the name of this structure. Yeah, that allow all these microtubules, yeah, all the spindle to form. So here is pretty easy. We're going to go through some of this terminology. Yeah, definitely the chiasma is not the right answer. This is the physical connection after crossover. Bar body, we're going to talk about that. That is related to X activation, as you remember. Centromere is actually the center here of the or this structure here that allows to pull the chromosome, but is not that related to the spindle. And cyclin is a protein that is involved in the cell division. Yeah. So the correct answer here will be centrosome. That will be those structures there. So I'm going to cover to define very quickly some of the other words and to show you that the best answer here is centrosome. So here you can see the drawing that I was trying to do. I wanted just that you, when you answer a question, you try to picture it so it can help you a little bit better. So here, what I was trying to show is this. Yeah, this structure that is located here. That is the one that produces all these spindles, and these are called centrosomes. And the centrosome is an organelle that is the main place where all the cell microtubules are organized. And here the microtubules are all these green structures that are going to pull the chromosomes. Okay, and here you can see a diagram of the centrosomes that are here. All the microtubules are here. Are these structures here that look like poles that it will help to um, pull the chromosomes. Yeah, so remember they are in charge of regulating the cell division cycle, and they are very important. Okay, so best answer here, it was the centrosome. Now let's go through some of the other terminology. Okay, so we're going to talk about now about chiasmata that we talked chiasma and chiasmata in previous question, but let's go over that and through some of other relatively similar terminology to define it very clear. So at this point, I'm sure that if I show you a picture in this picture, you know that here in the middle you will find the centromere, this region of DNA that have very few genes that have a lot of repetition, a lot of repeats of DNA, and here is where the microtubules will go and pull the chromosome. Yeah, so here is the centromere. So here the centromere, remember centromere is different from centrosome, and here you find this structure, that is the chiasma. Yeah, so the chiasma is the point of contact, the physical link between the two non-sister uh, chromosomes that belong to the homologous chromosomes. Okay, so remember centrosome, the one that produces the spindle. Centromere is this structure here that have a lot of DNA repeats that allow the microtubules to pull from there. Another terminology important here is this chiasma, that it will refer to the physical contact between a non-sister chromosomes, yeah. And in the chiasma, as we said before, is where the exchange of material can occur between both chromatids, and this process is called chromosomal crossover. Okay, so good. Now I wanted to show you this terminology. Let me delete this structure. And in the following sections of the chapter, and we already talked a little bit that, you will hear this terminology. Yeah, so we already talked about chiasma, but we have talked about homologous chromosomes, bivalent, tetrads, daughter chromosomes, and recombinant. So what I want to do now is just go over these definitions that they sound similar, and we're going to keep using it. So you know the definition now, so it's much clearer when we talk later. Okay, so first thing is homologous chromosome. Homologous chromosomes, you can see it here, and if you see them, yeah, they are pairs of chromosomes that they look approximately the same. Yeah, they look approximately the same in length. And you say, yes, they look approximately the same. The centromere position. And if you stain them, yeah, the G band patterns of the same genes, they will look very similar. Um, uh, one is inherited. Yeah, one is inherited from the mother. Yeah, okay, this was supposed to be a mother symbol, and here is inherited from the father. Okay, so homologous chromosome, pair of chromosomes that look very similar, one from the mom, one from the dad. Now let's talk about this one. So in the beginning, we have homologous chromosome. Now the chiasma was formed, and now you have bivalent. Yeah, so these are these bivalent chromosomes are defined as connected homologous chromosomes. They are connected. The physical point of connection is the chiasma. Okay, so it's relatively it's a related topic, but here the homologous chromosomes in the first section here they are non-bivalent. They become bivalent when the chiasma form. Okay, and now we have another terminology here that is the word tetrads. Okay, so we're going to compare bivalent and tetrads in the following slide. Okay, so well, before we were talking about bivalence in terms of physical contact. Now we're going to talk about bivalent, comparing it with a tetrad. The bivalent is one pair of the chromosome, the homologous chromosome in the tetrad. So here you will have 1, 2, 3, and 4. And the bivalent will be the one that have the physical connection. The tetrad, on the other hand, is the association of these 4 chromosomes. Yeah, and to be a tetrad, they only need to have at least one DNA crossover. Okay, so bivalent will be this one. The tetrad will include the entire 4. Okay, so you already know this is a homologous chromosome. In this case, the homologous chromosome, one comes from the mother, the other one comes from the father. They look relatively similar, but now there is no connection. Then they become bivalent when they start having a contact. Yeah, and tetrad is when you're not only referred to the ones that are in contact, but to all of them if one of them have contact. Now we're going to define recombinant and daughter chromosome in the following slides. So now, to finish, daughter chromosomes that we have talked about in the previous slide. Daughter chromosomes is the result of the separation of the sister chromatids. So here you can see what in the homologous chromosome you see the 2 of them. Here you see the separation, yeah, and here you see that the ones that come from the mother and the father are there, but now you see some of them that have a combination. And those ones are called recombinants. Yeah, so the recombinant chromosomes are the ones that experience genetic recombination during meiosis. Yeah, and this is important. In the following chapters, we are going to be talking about recombinants. Yeah, and we're going to talk about that the daughter chromosomes. They will look. Some of them will look like the parentals. Yeah, and this will look like a parental one. And there are other two. So from the four of them, two of them in this case will look like recombinants, and the other ones will look like the parentals. And the recombinants in the progeny, these will have a low percentage. Yeah, there will be very few that actually crossover took place, while the one that look like parentals, will have a much higher percentage. So, for example, if there is 1,000 progeny using fruit flies, depending on the location of the chromosome, you can have 2%, 5%, 10%. Yeah, so uh, you will have much less. Yeah, so from the 1,000 and 900 could be look similar to a parentals, while only 10%. Yeah, 10 between around 100 would look like the recombinants. Yeah, and we will cover that in the following chapters. But that is a concept that I want that you remember. Okay, so again, with this question, we review a lot of topics. We remember what it was a homologous chromosome, what is ambivalent, what is a tetrad, what are daughter chromosomes, and that the daughter chromosomes can be similar to parentals that have a lot of most of the progeny will look like the parentals, and there will be other ones that are recombinant chromosomes that are the ones that actually have where recombination took place. Okay, so now let's go back to the question. So I'm sure that now you know that the spindle pole is organized around a small region of clear cytoplasm near to the interface nucleus called, remember, close to the spindle pole, this small region is called centrosome. Yeah, so where the microtubules, we already explained that, is the centromere, is these regions where you have a lot of repeats of DNA where you pull the chromosomes, so this is incorrect. We talk about the chiasma, and this allows us to study, to review topics like homologous chromosomes, bivalent, tetrads, and the daughter chromosomes. Yeah, and you already know what is a cyclin. I'm going to show you that maybe you haven't heard about this expression, bar body, but I'm sure that you already remember that the correct answer is this. So just very quick, we will cover that in more detail later, but what is a bar body? So here, when we talk about the bar body, maybe you already read this in the textbook or you watch it already in the in one of the videos, that there is a process called X inactivation. Remember, in mammals, we have X in the sex chromosome. You have uh, females that have XX and males that have X and Y. So you have one chromosome expressing here and one chromosome here. In females, you have two X chromosomes, and you need to inactivate one of them. So one of them will not work. Why? To have a balance, yeah, a normal gene expression. And so how they inactivate it, and we will talk more in detail on you will later. But during development, one of them, both are active, and then one of them become inactive. Yeah, yeah. Here, the two chromosomes sometimes some of this one of them get inactive, and the other one can get inactive. So in this case, the blue chromosome get inactive, while in other cells the purple chromosome get inactive. But this is what happened during development, and in the chromosome, in the nucleus, and then staining will look like this. Okay, so this definitely, the bar body was not the answer. And I just wanted to remind you what it was about body. So remember, it's a small, densely stained structure in the cell nuclei of female mammals. Why only females? Because they are the only one that have two X chromosomes and contain uh, an inactive X chromosome. So this bark body is actually an inactive X chromosome and is a diagnostic test for femaleness. Okay, so definitely this is not the answer. And with this, we finished that question. I tried to bring several other topics to make that more clear. So if that question appears in the test, you will easily answer it. Now let's go to the second question. So now we're going to talk about the correct orders of the different stages of mitosis. And this is pretty easy. I'm sure that you remember this from high school. So it's this one, first prophase, then metaphase, anaphase, and telophase. And I'm sure that you have heard this, you already watched a video about that. So I'm going to go very quick. In the quiz, we cover this, all these sections, and we said that we are going to focus in this case for this question in this mitotic phase that is divided into 5 phases. Yeah, the end is the cytokinesis that is located here. That is the when the two cells separate. Um, how to remember this if you don't remember in any way is hard. Some people. To me, what happened is that every time that I teach a different class, there are things that I remember and other ones don't. So you might do the same thing you remember from the test and then you forget them. Some people use mnemonic, yeah, that they can help them for teaching. Sometimes it helped me because there is so much material. So some people use this. So for example, uh, this sentence, and you can use other one. Yeah, I prefer milk and tasty cupcakes. So it reminds you that I is from the interface, prefer prophase, uh, milk, and tasty cupcakes. Okay, if you already know, then you need to apply it, but in some cases, this could be useful for some people. So I just want to tell you that, and now let's move to the next question. Okay, this is a question that is very easy. Let's read it together. Regions of the chromosome with very few functional genes are called. So here, for example, the chromatids is something that we have covered. So when we have these uh, chromosomes, the chromatid, it will be one of this. We have talked that mid-repetitive sequence are sequences that are located, for example, in the centromere or the telomeres of the chromosomes, but this is not considered a region, yeah. And a nucleosome, so we know that the DNA, suppose the DNA is not tangled like this, yeah, it needs to properly be wrapped. So we're going to talk about these structures, yeah, that the DNA wrap around, yeah, so is properly organized, and it can turn into a chromosome after thousands and thousands of wraps, yeah. And these structures in blue are supposed to be the nucleosomes that we will talk in a second about that. So as a conclusion, nucleosome is incorrect, mere repetitive sequence is incorrect, chromatid is incorrect, and there is only two options. And I want to that you see that they sound very similar. um, Chromatin, yeah, both finishing chromatin. One is EU chromatin and the other one is heterochromatin. So the regions EU, yeah, this Latin EU means true, yeah. So this is the true chromatin. So is the chromatin that have a lot of gene, and the hetero, it means different. Yeah, so this will be actually the chromatin that have very few genes. It's not the chromatin that have a lot of uh, that has a the euchromatin has a lot of genes. Yeah, the heterochromatin doesn't have it. So let me show you and explain it in the following slide. So now you can see it in a much bigger schematic from your textbook. Here we have the heterochromatin in orange, and it's located in the centromere and telomere, that are regions that have a lot of repetitive sequence. Yeah, so they have very few genes. You don't need to have telomeres full of genes because, as you know, they can get longer or bigger. Yeah, and so their genes are not important. And here, the first place where things are going to break, the DNA could be in the centromere, the one that go through more mechanical stress because they are being pulled in the centromere. So you need genes there. The genes have to be in the euchromatin. Okay, so now let's talk. I'm going to show you a little bit more about talk about a little bit about the heterochromatin and the euchromatin at the microscopic level. So do you remember that I was talking about the nucleosomes? So here you can see in blue each one of the nucleosomes, yeah, and in black here this is our friend the DNA that is there, that is being wrapped in each one of these nucleosomes. And the nucleosome have histones that we are proteins that are in charge. If you see, there is two turns, and we will talk about that. So one turn, and there is another turn, and this allow the proper packing of the DNA. And I want that you compare right now the euchromatin and the heterochromatin. So one, here it says that is the heterochromatin is silent, while the euchromatin it says that is active. So why do you think that one is active and the other one is silent? I think I'm sure that you saw the space. Yeah. So here, oh, as it's active, you can have enzymes. There is a space here for an enzyme to locate here and start transcription. Yeah, you can also have if other proteins like transcription factors that they can locate there, and they can start the process of transcription. So this is what they are active. On the other hand, when these enzymes are going to try to find a place here, there is almost no space. So they will go to the regions that that allows them to have this complementarity, yeah, to locate here, to attach, and to start transcription. So in our words, I told you about two different type of chromatin. One that is the euchromatin, and we said that this one is slightly packed, yeah, and here I showed that it was formed by DNA that, as is slightly packed, later it will be transcribed into RNA, and then it will be transcribed into proteins. So it is enriched in genes and is often, not always, under transcription. Okay, so they are very active regions, and they are under transcription constantly. On the other hand, you have the other regions that are the heterochromatin, that are the ones that are in this question. And when you do the stainings, the G-band stainings, all the different stainings, these are dense material. Why dense material? Because they are fully packed, So as they are close to each other, they look more dense, and they are generally found in the centromere and the telomeres. Okay, so we talk now. You know what is a euchromatin and what is a heterochromatin. Now let's go over with some schematics on the other questions. Okay, I already tried to explain this and the schematic before, but I think this is much clearer. So here you can see that there is a chromosome, and each chromosome here, they are formed by two chromatids that, if you see, they are very similar because they just replicate. So they form here, and they are fused through this, they're together joined in the centromere. Okay, so a chromatid is one of the identical half of a chromosome that has been replicated in preparation of cell division. And they are linked together. These sister chromatids are joined to a constricted region called the centromere. Okay, so definitely chromatids is not the answer. Euchromatin is not the answer. Mid-repetitive sequence is not the answer. And we know that nucleosome is not the answer. Now I'm going just to show you a few diagrams of the nucleosome and talk a little bit more about the nucleosomes. So we generally talk about chromatin, chromosome, chromatids, but hardly ever we go in detail and look at the microscopic level to talk about DNA. When we do that, we know that the DNA is very well organized, so it doesn't tangle, and is organized thanks to the nucleosomes. Here you see a nucleosome that is formed by different proteins. So here you have 1, 2, 3, and 4 proteins there, and here there will be a fifth one that we will talk in a second. Each one of those are histones, and if you see there is one turn, yeah, and there is another turn. So it always gives 2 turns. Here you see one going here and the second one, and here there is like a pin here that allows to attach properly. Okay, so a nucleosome is the basic repetitive unit or a subunit of chromatin package that is inside of the cell's nucleus. Okay, so definitely here we are not talking about very few functional genes, many or few. This is just something completely different. Okay, the nucleosome is something completely different. Okay, I hope that now you understand why when we talk about the chromosomes, the specific regions, there are two, the euchromatin and the heterochromatin, and the euchromatin has a lot of genes and the heterochromatin have very little number of genes, yeah, or functional genes. Yeah, they are formed by more by repetitive sequence in the centromere and telomere. Okay, so hopefully now this question is pretty easy for you. Okay, so I gave a long answer, yeah, about this question about heterochromatin. I wonder you think about this. I have been talking about this type of chromosomes, yeah, but what about prokaryotes? Do prokaryotes have telomeres? Do prokaryotes have heterochromatin? Do prokaryotes have centromeres? You can stop the video and think about it. Yeah, I have been talking a lot about that, but this is the common chromosome that we see in eukaryotic, yeah. But what about the prokaryotes? What happened there? Okay, so here I just wanted to show you this very interesting debate that we wish that this story was much simpler. But the idea is that when we think about chromosomes, yeah, we generally look at this in the eukaryotes. There was replication, and you have these two chromatids for the cell division, and here both will have. Let's put it in black here. The telomeres that are very important, yeah. And as you watch in the video, there is the Hayflick number, so there is a limit of number of cell division for cells before they go to a process similar to apoptosis. And the question is like, could aging and sex could be linked? Yeah, so we needed to mix our chromosomes, yeah, and we need to create telomeres and centromeres to do this kind of things. However, in our organism, like prokaryotes, many of them are asexual. They don't need to mix the chromosomes. Yeah, in that way, the chromosomes are much smaller and they are packed with DNA. So they are circular. Here you don't need a centromere. Here you don't need um, a telomere. So, and definitely the question is, do they have like indefinite proliferation? And maybe aging could be related to some degree to the presence of telomeres and the presence of these linear chromosomes. And it's an interesting debate that I just wanted that you think about it. That we generally talk about this, and we have been talking about telomeres, centromeres, and heterochromatins, but the prokaryotes don't have those things. They use a completely different mechanism to create diversity in their genome. They use conjugation, they use plasmids, and they are transferring that. We In our case, we cannot transfer plasmids. Yeah, no, you are not going to get a DNA from another person through a plasmid. The bacteria are the ones that can do that, as we have seen in previous chapters. Okay, so now let's summarize the idea of heterochromatin and euchromatin. So here I have this table that summarizes all the information. For example, euchromatin is found in prokaryotes and eukaryotes, but heterochromatin is only found in eukaryotes. Euchromatin is active, as we said before, while the heterochromatin is inactive. The DNA density here, there is a low DNA density. Here you have a high DNA density because it's all packed, as all the nucleosomes are all close together. And there is, so here is loosely packed, and here is tightly packed. Okay, with this I want to just have a point of discussion to make it interesting, that you think a little bit about biology, and now we can continue to the next question. Okay, so now the question is, which type of histone is not part of the nucleosome core particle? And here you have all these H1, H2, all of them that stand for histone. So I want that you think all the things that you have read from the textbook or that you in the video lectures and try to answer this question. So you can stop the video now. If not, I will answer it in a second. Okay, so here it's pretty easy. The answer will be H1. Yeah, I just remind you, the nucleosome is this basic unit formed by histones. Yeah, those histones are named here. So the H2A, H2B, 3, and 4. Yeah, and here you find each one here, the histone, here you find the linker, and all this structure is called the nucleosome. However, in addition to the core histones that are located in the nucleosome, you have an additional one that is the histone H1 that is located like a pin outside of the nucleosome. So, which histone is not is not part of the nucleosome? Pretty easy. Here is shown the H1. The other ones, all the other ones that are here, are part of the nucleosome, so they are incorrect. You can see them here. Okay, we're done with this question. Let's move to the next. Okay, so this one is pretty easy. I'm sure that you remember this from high school. Let's read it together. The mode of cell division that results in 4 genetically different daughter cells, each one containing one haploid of set of chromosome is known. So key information here, 4 genetically different daughter cells. So this just means that this is not mitosis, and that each contain one haploid set of chromosome. This means that is meiosis. Right away, cancer will give so much variation. It will not give 4 cells. It could give many more. It could give not an haploid. It can give so many other things. And the other interface doesn't create cell division. Somatic cell regulation don't make any sense. So here, pretty easy. The cells go through their interface. You go through the process of mitosis, and you get 1, 2, 3, and the 4 different daughter cells. And as there was here, if you remember, when they go from meiosis 1 to 2 in prophase 1 in the 5 subphases, there is a recombination. So if you see each one of these daughter cells, of the 4 daughter cells, will be slightly different. Okay, and they have half of the chromosome, so they have a haploid set of chromosomes as is shown there. Okay, so pretty easy. Now let's move to the next question. Okay, so let's read this question together. Again, we're going back into some type of inheritance and pedigrees and crosses. And I think it will be hopefully pretty easy for you now. So here, let's put it in maybe highlighted in black. So which of the following type of inheritance, so let's highlight here with yellow, this is a key part of the question. They're asking for type of inheritance, this is the question. And they are asking is characterized by, and here they give you another important part of information, by an affected father, and here who has affected daughters but not affected sons. Okay, so here they give you three pieces of information that there. Here you can see the question. They're asking you what type of inheritance, and to answer this question, they tell you the father is sick or is affected. The second thing, the daughters are all affected, and the sons are not affected. So, what type of inheritance do you think that it is? Is an autosomal or is an sex link? X link, Y link? It is recessive or it is dominant? Okay, so let me show you with some schematics to make it easier for you to understand. So here, the key to understand this question is to analyze all these three sources of evidence or just statements that can help you to answer the question. The answer is it will be an X-linked dominant. And why it is an X-linked dominant? Because here you have, in this case, the father is affected, yeah, and the mother could be affected or could be unaffected. They don't include that information, so it's not important, yeah. So what happened here is that if you see it, you have two chromosomes, one that is the Y one that is going to go into the males, into the son, into the progeny of the next generation, but this will be unaffected, yeah. So the gene that is that is in charge of this type of inheritance is not in the Y chromosome, so it's not a Y linked. So this is gone, yeah. And you see that the daughters are the one that get sick, only the daughters. So it doesn't seem to be autosomal. So there is two options: the X-linked recessive or X-linked dominant. And if it was recessive, it will need the two, the father and the mother, to be affected. It will need the two of them. However, you remember the mother, it seems to not play a key role. They don't give any information if the mother is sick or not. So the most likely, based on this information, is that is because there is an X-linked dominant. Yeah, all the daughters are sick because he has one X chromosome that have the mutated gene that is transferred to all the daughters. Okay, so all the other options seems to be wrong, and based on this information, only this information is correct. Okay, and I hope that it was clear. And if you try to, you can play with other type of answers, yeah, and you can see that they will not fulfill they will not fulfill the statement, and the best answer based on the information on these just two lines of information, it will be answer E, X-linked dominant. Okay, I'm being super repetitive here, but hopefully you remember because you have heard so much about mitosis. But here what we want to add a new section in this topic, that is the topic of crossover in the mitosis, and the crossover takes place in the prophase 1, which consists of the following substages in order. Okay, so you need to see which one is the first, which one is correct of this ones. Okay, so here just kinesis means that you cut something, that you break something, so for sure it should be one of those trees. And now you have to decide which one of those, A, B, or C. Yeah, and if you're like me, this sounds like complicated, but maybe we can find a way to make it easier for you. So let's review this very quick. The correct answer will be B. Uh, if you remember, leptotene is where the chromosomes condense. Zygotene is when the synapses take place. So the synaptonemal complex, so the proteins that are going to align them are there. And in pachytene is where the crossover takes place. Yeah, and here you start mixing, exchanging the information. In the end, diploten is when the these synapses complexes start to disappear, but you can see the contact, physical contact that are the chiasma, and here in diakinesis is when the chromosome contracts, and already you can see already the crossover there, and they are ready to move into the next step of the meiosis. Okay, so but now a question is, we know that is B, that they have different sections. Again, how to remember this? Uh, Difficult to remember so much information. In my case, I try to use mnemonic. So try to find the best way to remember for you. In my case, it helped me to use to think about, I don't know, Love, Zootopia, Police Department, and Dragon. This for me is much easier to remember all these names. Yeah, so I remember that this uh, the initial of these ones. Yeah, and in that way I can remember. You can find a different way. Just remember LZPDD, something that helps you to remember, so you can remember them in order. Okay, at what substage of prophase one does crossover take place? So the question, they're asking you a substage, and here you can see the five substage, and I'm sure that you already know the answer. I have been repeating that multiple times. So I'm sure that you know this pachytene. Yeah, that is when the crossover, the DNA exchange by the non-sister chromatids, and you can see it here. Okay, so here all the things they are exchanging. So you go from the synapses, yeah, into the crossover, and then you form the recombination. Okay, so pretty easy. The diakinesis, wrong because it's the end. Leptotene is when these chromosomes start condensing. Zygotene is when the synapses start, and diplotin is when you can see the chiasmata, but the synapses complex start disappearing. So the only answer correct is the pachytene. Okay, so now another question related to crosses and inheritance. So let's read it together. Um, okay, which of the following is not? And I'm going to make a box here. Remember, not a characteristic of a recessive sex-linked inheritance. Okay, so we need to remember what is a recessive sex-linked inheritance. So remember, so it has to be X and Y, and um, or it can be, so it can be in yeah, and females or males, and it has to be recessive, yeah, independent of each one of those. So they give you all of these options, and you need to think about it. So think about it, and in the following, I give you the answer. Okay, so now let's go through each one of the questions, of the answers. Yeah, we know that C is the correct one, and I will have a diagram, this diagram bigger there to explain why this one is correct. But let's go over each one of these. Reciprocal causes give different results. No, we know that if it's a sex-linked trait, independent if it's dominant or recessive, reciprocal crosses will always give different results. So this one is true. So this is wrong because we're looking for the false one. More males are affected than females. Yes, this is also true. Why? Because because as males only have one X or one Y, they are more likely to be affected. Yeah, they don't need the two of them. Female will need, for example, 2X. And the female carriers can produce normal and unaffected sons. That is true too. So they will need the two chromosomes, they need one, they can transfer the sick one, yeah, or they can produce um, as they are carriers, yeah, so they can have transfer or the sick, or they can transfer the unaffected trait, yeah. So this one is true. Affected males may produce affected females, and this can be also true, yeah, if both mother and father are sick. Yeah, so that is all of this one are true. The only one that is false is this one. Okay, so this one is the one that is actually the answer. Affected males can produce only affected sons. So let me show you why. So pretty easy. Here you see an affected male, and it says it can produce only affected son. And here you see an example that is this is wrong. Here you have an unaffected son. Why? Because here they transfer the X chromosome from the female, and the Y chromosome, it doesn't go, that is affected, it doesn't go to the male. It goes the Y chromosome that is not the one that is affected. So very clear, this one is wrong. Okay, with this we finish many of the questions, many examples of questions that they can be in the test. If you have any doubt, just let me know. Have a good day and bye.